everyone. It's three. Hello, let's say hello to everyone. It's 3:03, so it's time to have fun with RNA. Of course, we have to thanks to all of you being here, to all the attendees, because without of you, no webinar can be done. This is a webinar of RNA Collaborative. Two important institutions, MD Anderson and UCSC, are presenting, I would say, some very interesting results and research on RNA and non-coding RNA. And let me to introduce first Anca Jurj, who is a postdoctoral fellow at Translational Molecular Pathology in MD Anderson Cancer Center. She is a very hardworking postdoctoral fellow. I can tell you that she's working in non-coding RNA center, led by Anil Sood, myself, and Gabe Lopez-Berenstein on several projects. And let's have Anca to start the show with RNA Therapeutics. It will follow by Dr. Manuel Ares on a very interesting project on polyamine-responsive riboswitch. But let's first start with RNA Therapeutics. Anca, thank you for your talk. And again, thank you everyone for being here with us. Good luck, and let's have fun on RNA. Thank you again to all. Thank you.
Hello, everyone. Thank you, Professor, for the kind introduction.
Okay, so thank you, Anca. Now let's go to the next speaker because time is money. Thank you very much, and I'm curious for the next presentation. Thank you.
Okay, thank you, George. Hello, everybody. My name is Michael Stone. I'm a professor here at UC Santa Cruz in the Department of Chemistry and Biochemistry, and I'm also a member of the UCSC RNA Center. It is my pleasure to quickly introduce my colleague, Dr. Manny Ares, who is a professor of molecular cell and developmental biology here at UC Santa Cruz. As many of you will know, Manny is widely recognized for his extensive contributions to RNA biology, and with a particular interest in understanding mechanisms and regulation of pre-mRNA splicing. Over the years, Manny's lab has been at the forefront of using a variety of technical approaches to dissect splicing regulation, from systems such as yeast all the way to human, like we'll hear about today. And Manny's really been, and his lab has been a key figure in shaping how we think about RNA processing in a more general and evolutionarily conserved way. So I'd also like to quickly mention that in addition to his extensive scientific impact, Manny has a really extensive track record as a dedicated mentor and educator, and is known still as a source of curiosity and for training rigorous scientists that are working all over the world. So it's my great pleasure to introduce Manny today, who will tell us about recent work from the lab looking at an ancient polyamine responsive riboswitch. So Manny, take it away.
Thanks, Michael. Let's see, share my screen. And then start my presentation. Good? We can see it?
All good.
Anyone? Yeah, yep. Good. Okay. Yeah, so talking to you from Santa Cruz, welcome. Thanks for listening. This is a shot of our campus. It's the part in the lower left there that's all in the woods near the ocean, and near the little town of Santa Cruz, which is an awesome place to visit. Please come and see us sometime, especially if you're interested in RNA. I'm going to open with an introductory slide that's a little dense for most introductions. Don't be alarmed. I just want to familiarize people a little bit, if they're not familiar with the amazing set of molecules that we call polyamines. They're a metabolite of cells. They're something that most cells make, almost all cells make them. They're positively charged at neutral pH, and they're tightly regulated by almost all cells that have them. Their levels are tightly regulated. And one of the ways we know this is that many cells have a gene called antizyme, which is an inhibitor of the enzyme at the very top of this biosynthetic pathway for polyamines called ornithine decarboxylase. Antizyme binds to ornithine decarboxylase and shuts it off. And almost all antizyme genes have a frame shift, a +1 frame shift. Their genes are actually broken. Their coding region is not complete, but they require a +1 frame shift. And that +1 frame shift is triggered, enhanced by high levels of polyamine, possibly through direct interactions with the ribosome. The idea being that if polyamines get too high, you do the antizyme frame shift, make more antizyme, and turn off the pathway at the top in a very direct way. And in addition, there are a couple of other genes. We'll talk a lot about SAT1, but there is an antizyme inhibitor whose regulation at the level of splicing also acts to control polyamines. This gene here, SAT, we'll talk about more, is activated in order to excrete polyamines when polyamines become high. And so I'll just now kind of dive in and show you the gene, SAT1, that stands for spermine acetyltransferase. And what I'm showing here is the UCSC genome browser shot of the two isoforms of SAT that one observes. This top one, if you know the browser hieroglyphics, there's a complete coding region. The fat exons indicate that. And so that's the productive mRNA form. And then there's a second isoform that has this extra exon, and you can see all the exons are skinny. That's because this isoform is subject to NMD decay, nonsense-mediated decay. And we know nonsense-mediated decay is around to get rid of, sort of bad splicing products, but this exon is highly conserved. In fact, it's more conserved even than the coding exons on either side of it, and yet it doesn't code for protein. It must be there to help regulate the gene, or it must be there for some evolutionarily advantageous reason. And so what we started to study this, because we've been very interested in the association between alternative splicing and NMD and how that a coupling of those processes can control gene expression post-transcriptionally. And so we looked at the other evolutionary characters in this sort of comparison of different organisms. And you can see by these asterisks in this alignment that this exon, which is shown in pink here, is very highly conserved. And over here in the lower right, I'm showing how we normally assay this splicing event. Because the include isoform, which is shown, this larger RT-PCR product is the include isoform. In normal cells, you don't see very much of it because it's subject to NMD. This is the mRNA. However, if you block NMD with emetine, a translational inhibitor, since ribosomes have to be translating in order for NMD targets to be decayed, we inhibit translation and we stabilize that isoform. And so actually, this lane may show more accurately the alternative splicing distribution. That is, most of the transcripts have this exon included, but they're being rapidly decayed. Okay, and then if we add either of these two drugs that perturb polyamine levels, we can see now in the presence of emetine that the alternative splicing is changing. And what's happening here is this BENS, this is a bit short for benzspermine, is a non-metabolizable polyamine analog that mimics high polyamines in the cell. And so that triggers, BENS triggers exon skipping and promotion of synthesis of SAT, which I told you previously is required to excrete the polyamines from the cell. So in response to high polyamines, alternative splicing switches to make mRNA so that a protein that reduces polyamine levels can be made and polyamine levels will go back down. Conversely, if you inhibit the top of the pathway, this is an ornithine decarboxylase inhibitor, difluoromethylornithine, that promotes alternative splicing to include the exon and reduce SAT levels, which is what you would want if you're depleted of polyamines. You would want to block that enzyme from excreting so that polyamines come back up, and you're in a homeostatic feedback loop here to maintain tight control of polyamine levels. Okay, so I told you that there's these four we're going to look at more carefully: zebrafish, xenopus, human, and mouse. This is the human right here. And so what Jenna Lapointe did was get cell lines for mouse and xenopus and zebrafish and show that in those endogenous cell lines, the regulation of the corresponding SAT gene is conserved. And then kind of in order to test the evolutionary, robustness of what we're looking at here, Jenna transplanted into a reporter construct each of the exons plus its adjacent intron sequences into a splicing reporter, and then transfected each of them into mouse N2A cells. And what you can see is that the regulation, so this is now exon inclusion being inhibited by the addition of the high polyamine analog, the polyamine analog. So the regulation is preserved across more than 400 million years of evolution. And I would also note that that the temperature is different here, right? These are cold-blooded organisms, and these two are warm-blooded. So we're excited about the fact not just that this is an evolutionarily conserved element of some kind of regulation, but that it's conserved across, it's functionally conserved across many millions of years of evolution. Okay, so what is it? So when we line these up with an eye towards looking at RNA structure, because the exon is, I might have not mentioned this, but it's highly conserved and it's not coding for protein, what is it making? You can see right now that we've drawn it in this kind of stem loop structure here on the right, but that if you investigate the alignment here, you can see there's very little in the way of phylogenetic variation that would support the evolutionary evidence for a stem. In other words, what you want for an RNA structure to evidence for its conservation is sequence variation that preserves the secondary structure. And we don't have that sequence variation. So we can't really say that this is the correct secondary structure or that it's the only secondary structure. But because these guys down here, the frog and the zebrafish, are the most different, we decided to study all of these kind of together for a while. But the main points here are insufficient variation to prove the structure. And there's the 3' splice site of the exon is here. I didn't point that out in the 5' splice site. That's the complete exon right there. So it overlaps this intriguing RNA structure. And because we're sort of in the mode of thinking, as I told you, that maybe that regulation is direct, we went looking in the PDB for high-resolution RNA structures that might also contain a spermine or spermine molecule. And that might tell us something, we thought, about how this RNA might interact with polyamines. And these are the examples that we pulled out. And I'm putting this here to show that basically we couldn't figure anything out from looking at this because the spermine is found in association with different RNA structures in very different ways, right? So spermine has 4 amine groups that are positively charged. And so they're spaced out by different methylenes, and sort of they're tailor-made to interact with the negatively charged phosphate backbone, which is why they're actually included often in these crystallization mixes and in these structural buffers for structural analysis. But here, just to point this out, there's 2 tRNA structures here, and here's spermine sitting right here in this particular tRNA, and this one, it's over here. This is a group 1 intron, and you can see yet a different way. So we didn't get anything from that, but we're still, in terms of specific ideas, but we're still excited about the possibility that this is a direct regulation by interaction between the cationic polyamine and an RNA backbone. So without a phylogenetic evidence for this structure, we turned to structure probing methods. And in addition, we wanted to understand whether spermine would change the structure, the folded structure, or the folded structure might be different with or without spermine. And this is an experiment that Kinneret-Sheffer did, and it's very simple. It just has RNA in a test tube. These are T7 transcripts that we make. And you either add spermine or you don't. You heat the RNA to 95 and quick cool it and add some salts. And then we'd use the shape, this acylation of 2' hydroxyl groups as a chemical probe for RNA structure. And I think you can see on the right that when you fold the RNA with and without spermine, you get two really different structures. One of them, I can't see it because my face is sitting on top of the image, but I know what the gel looks like. This lane here has reactivity in what we're thinking is this loop up here and is protected along the sides here. But then in the presence of spermine, that loop becomes protected. It's no longer reactive, and a region here becomes more reactive. And so this gives us a kind of an idea that perhaps spermine is allowing the RNA or helping the RNA form into a pseudoknot where the loop comes to pair. You see these C's up here might come to pair with these G's across here and push this strand out, and that might account for that the structure change that we're seeing. So we might be seeing a structural switch between a simple kind of stem loop and a pseudoknot. So does this also happen for frog and fish? I told you we were going to take an evolutionary stand here. And so what you can see, I think, is that the frog does this switch as well, and the fish does it as well. And so these are the models now, structure models for, even though there's sequence variation in these regions, these two RNAs, which are different from each other and different from human and mouse, all four of these RNAs now are able to do this same flip with polyamines. Okay, so these are primer extension gels with radioactive oligonucleotides. This is old school stuff. You know, nobody does this anymore. Everybody does, RNA, they do sequencing libraries. Instead of using a reverse transcriptase that stops at the modification, you can make an amplicon, because you can have a reverse transcriptase like the Tegert RTs that will read through, or superscript 2 with manganese, that will read through the modification and insert an incorrect base. And then you make a sequencing library from those products, and you can mark where the mutations are. And then those sites of mutation you can infer were modified, and then you use those modification patterns to infer the structure. And so we did that now for the human, and this is in vitro again, and I'm showing you the same thing that basically we just showed you with the old school gels, which is that when you have fold without polyamine, the loop is exposed, and then when you fold with polyamine, you get what we're going to call the pseudoknot helix that you're seeing up here, and that strand gets displaced. Okay, so what does this mean? How much polyamine are we talking about? RNA is negatively charged. It might not like to fold unless you shield it with some positive charge. Maybe this is non-physiological. Maybe this is just, some kind of chemistry thing. So to challenge that notion, we titrated the amount of spermine required to make this flip. And what you can see here is this is the concentration of micromolar of spermine that is in the reaction when the RNA is heated and then cooled. And what you can see is somewhere between 10 and 50 micromolar, the structure flips from the no stem loop, no spermine or low spermine stem loop form to the pseudoknot form. And just to give you perspective, spermine in mammalian cells is about 1 millimolar. Okay, and so this is, now not all of that is free, probably a lot of it's bound to proteins and nucleic acids, but 50 micromolar is not a lot compared to physiological levels. So we think that this titration means that this RNA is sensitive to physiological levels. It seems it makes it less likely to be just like a chemical shielding effect. And consistent with that, we tried all kind of additional polyamines besides spermine. Spermine here again is that protected loop, reactive loop. You can see that spermidine, putrescine, ethylene diamine, and we've tried lots of other things there is specificity for this reaction. Spermine and some of the larger other polyamines will promote this structural rearrangement or structural fold, whereas the smaller ones do not. Okay, so we think it's physiological. We think it has specificity. I want to tell you a couple more things about this because it's sort of interesting. If you add spermine at low temperature, it does not trigger this. You have to do the boiling thing. You have to do the 95 degrees. If you fold it and then add spermine, so you still get the loop. This is a stem loop form. If you add spermine, fold, and then remove the spermine afterwards, you stay in the pseudoknot form, and magnesium doesn't really do anything. So what this means is that somewhere in the folding pathway above 37 degrees, there's some place where the spermine is having its effect during folding. And to kind of investigate that more fully, we did this experiment, which is a little complicated, but not too bad. We take the RNA and fold it without spermine, and then we add spermine and heat to various different temperatures to see how high we have to go in order to get this pseudoknot to form. And what you can see here is that at 65 degrees, we still fall back into the stem loop. But if we go up to 80 degrees, we can obtain the pseudoknot structure. So what this means is that, okay, when does the cell ever get to be 80 degrees or above 65 degrees and be alive? Well, never. But there is a time when RNA is unfolded, right? Which is sort of right after it's born. And so what we think is happening is that the nascent transcript might be coming out of RNA polymerase in an unfolded state. And at that moment is when spermine is sensed. So this is our model for this with nascent RNA. This is RNA polymerase going down DNA template, making a nascent RNA, which is unfolded as it comes out. Some initial folding events might happen, and the RNA is still sort of undecided about what it's going to do. It might encounter spermine, which sends it down a pathway to the pseudoknot, which promotes skipping of the exon. Or if spermine is not encountered, the folding persists, continues along this pathway, leading to the stem loop. And now the exon is included and the mRNA is destroyed by NMD. Okay, so how would we test this? Well, it turns out that Sylvie Ruskin's lab has been working on questions that are quite relevant to this, and they've generated an absolutely wonderful set of programs called seismic RNA that is useful for deconvoluting mixtures of RNA of RNAs that are in different structural states. So we're talking now not about I have my RNA, what is the structure? We're talking about I have my RNA and it has 2 structures, 3 structures. Those structures are all important for the function. How would I be able to tell in a mixture how much is folded in one way or the other? Okay, and I'm not going to go through all the details of this right now, but they've been super critical in helping us get at these questions now. So what we were asking now is there evidence that the pseudoknot folds more frequently in cells now, not in a test tube, when we jack up polyamine levels in the cells? And so we treat the cells with the polyamine, high polyamine conditions. We use dimethyl sulfate as a probe. It modifies A's and C's on their Watson-Crick faces, and this gives us structural information. We use a reverse transcriptase that puts mutations in where the DMS modifications are, and we get the sequences, and then we run this through the seismic RNA where the reads are analyzed, they're clustered to where they have correlating different changes, and then you can use the clusters to independently predict the two different structures, okay? And so this is what the experiment looks like when we have no polyamine addition and we run this. We find that about 92% under the no polyamine conditions have reactivity in that loop. That's sort of the key thing that we can see. And some 8% of the reads are in some kind of other state that we don't know what that is exactly. But when we treat for 30 minutes under high polyamine conditions, we find that a substantial fraction, more than 50% of the RNA now has a reactivity profile that looks like the pseudoknot, and another 42% looks like it's the stem loop, just like when there's no polyamine. So what we conclude from this is that benzspermine seems to be inducing a subpopulation of the pre-mRNA. Our primers for these amplicons are in each intron on either side. So this is pre-messenger RNA we're looking at. And the high spermine conditions cause the pre-mRNA to fold differently. So here's the model. Polyamines are shown as these little pink asterisks. And here's the happy cell with perfect levels of polyamine. And some of the nascent transcripts are folding in a way that will make the stem loop and cause them to be destroyed by NMD. A few others are making the pseudoknot, which will cause them to make functional SAT mRNA, and that'll help. That'll do what that enzyme does. But stochastically, if at some point polyamine levels drop, then you stop making nascent transcripts that have the pseudoknot because there's not enough polyamine. And now all of that becomes included and destroyed, and you prevent SAT from lowering polyamines. Conversely, if stochastically or under whatever situation polyamines become high, now you activate the formation of the pseudoknot, and many of the nascent transcripts end up being mRNA, and you can lower your polyamine levels back homeostatically. Okay. Do I have time for one little more thing? I'm going to do it anyway, just see what happens here. Okay, so we're very curious whether there's other places in the human genome where there might be this same kind of situation where we have splicing associated with NMD and associated with highly conserved intron sequences that might represent sensors of small molecules like polyamine. So we did an RNA-seq experiment under the kind of conditions that I've been talking about here, where we trigger high and low polyamines and block NMD. The expression, this is just a sanity check. The expression of the polyamine regulating genes that are supposed to go down. The ones, including SAT, that are supposed to go up. And that's sort of shown here on the pathway again. This demonstrates what we're seeing with, this is SAT was what we've been talking about. Here's azin, another polyamine enzyme. And you can see that in this case, it's regulated in the opposite direction. You activate a 3' splice site. Again, a highly conserved intron region. We don't know if this is, what the structure of this is yet. We're still thinking about that. Another set of genes that's very interesting, the click kinases. Here's an exon. They're orthologous genes. This is the orthologous exon in each case. Again, highly embedded in highly conserved intron sequence, and then high polyamine activates inclusion, which activates these kinases. And these kinases, one of their well-known functions is that they activate, they phosphorylate splicing factors such as SR proteins to activate them. And when we look at SR proteins, we also see them responding to these polyamine perturbations in a way that looks like they are being activated as well. And again, alternative splicing embedded in ridiculously highly conserved intron sequence. And so we're very curious, about what that is. So here's the summary, the take to work message. We have this highly conserved non-coding so-called poison exon that is included to control SAT mRNA production. It responds to polyamines. The folding of this exon responds to polyamines in vitro. It's the response to polyamine is physiologically in the physiological range, somewhere high in the folding pathway, but we don't really know if the polyamine stays bound like a traditional riboswitch might bind its molecule. In vivo, we see evidence that polyamines are altering the structure, the folded structure of the pre-mRNA, and we propose that the pre-mRNA spanning this exon directly senses spermine and folds co-transcriptionally to regulate SAT in order to maintain polyamine homeostasis. And then there's other suspicious looking sequences out there in the transcriptome and the genome. And these are the amazing folks who did all this. Jenna is my lab manager and not only does all these amazing experiments, but keeps the lab from disintegrating almost on a daily basis. These folks initiated the project actually some years back now. Julie Nee first discovered this when we were searching for alternatively spliced pairs of isoforms, one of which was NMD sensitive. Kinneret Sheffer really made the breakthrough of being able to show that polyamines affected the folding in vitro. Pahlavi characterized much of the biochemistry. Nick and Michael have been helping us with in vivo probing. Haller, I didn't talk about all the mutations we made in this structure. Haller did a lot of work on that. JP helps us with our computers. And then Sylvie Ruskin's group has been super helpful, introducing us to the proper use of this seismic RNA package for deconvoluting the structures that we see in vivo. And I will leave it there and take questions. Thank you.
Great. Thanks, Manny. That was awesome. There are a number of questions, so I'm just going to dive right in.
Yeah.
So the first question was very interesting. I was wondering, since most of the bases do not really appear to change their reactivity in the probing of the stem loop upon addition of spermine, can it be that the protection you see in the loop is just because that's where the spermine binds to the RNA?
That's a great question. I mean, I suppose it could be. We have mutations that disrupt the protection. And we have done some probing on some of the mutants, but I like the spirit of the question. I don't know where the polyamine binds, and I threw my slides away. But when you look at the different pseudoknot structures, what you see is the strand that's displaced has a conserved sequence, and my suspicion is that actually that it could be the spermine somehow interacts with that short. It's like an 8 nucleotide sequence that's present in all of them. And that maybe that is what prevents the stem from completely folding and allows the loop to access the former partner of that strand. But, we are super interested in what the structure of this thing might really look like at high resolution and whether there is a spermine actually bound to it. Great question. I don't know the answer.
Okay, thanks. So I'm going to move on to the next one. Instead of refolding the full-length RNA, have you tried to probe the co-transcriptionally folded RNA, for example, by performing T7 transcription plus minus spermine, or co-transcriptional shape map-seq, or SPET-seq might be a cool way to look at it?
Yeah, so we have wanted to try to get more in vivo specificity on the nascent transcript, like pulling down. There are a number of methods where you pull down. You either capture the nascent transcript or with polymerase as a ternary complex, and then you probe that. We haven't gotten to that level yet. We did try an experiment in vitro with T7. So those of you who do T7 transcription know you put spermine there's a lot of it. Actually way above what you need. And so Jenna did an experiment where you have to lower the spermine, have to take it out of the reaction. And the transcription yields are lower, but we think in an initial experiment that we did see something even with T7 that made it seem like we were starting to get the the folding to be spermine sensitive, but it's hard because the transcription is also changing quite a lot with T7. Rather not do with T7 anyway. So, but yes, this is a great direction to go, and I would love to do that.
Great. So there are a number of questions that I think I can combine, asking about something that you mentioned, Manny, the use of mutants. So have you tried to probe plus minus polyamine mutants that disrupt the structures? And have you looked at compensatory mutants? And then another question sort of asks something similar about, are there have you tested mutants that perhaps disrupt genetic function? I think trying to get at sort of the connection between the function of the model and structure. And so maybe you can comment on your use of
Yeah. So a couple things. So we have done those kind of classic, break one strand, break the other strand, and then recombine the double, design them so that the double mutant restores the strand. And it's a little complex because that structure actually is enforcing a rate of inclusion or skipping, right? In other words, the structure itself imposes some level of exon recognition, irrespective of the polyamine condition. So that when you mutate it and you open the structure up, suddenly now you get a lot more inclusion. And then when you close it back up, you knock it back down. So there's two things that are happening that we're trying to separate with the mutants. One is how does the mutation change the basal level of inclusion? And then is there regulation still present on top of that? And we do see that the structure is important both for setting the correct level of inclusion and for the regulation, although the lower parts of the stem are not as important for the regulation. But the other thing it says about that is that there aren't primary sequences embedded in this that are doing a lot. And I'm not ready to say that we know everything, understand everything about the mutations that we're making. One thing we tried to make was mutations that would force the pseudoknot to happen, and that we weren't able to really convince ourselves that those mutations were actually doing that. So I think the spermine is playing some really specific role in it. But yeah, we have that is the strategy we took, and it's complicated by the fact that the structure is also defining a level, the basal level, as well as contributing to the regulation.
Great. Thanks, Manny. So there's a couple more questions. We'll try to get through them. They're interesting. And I guess whoever is in charge can cut us off whenever they're ready to do so. So Niels Walter asked the question, says, great talks. Spermidine often competes with magnesium for similar binding sites. Would varying intracellular magnesium levels have an impact on the hairpin pseudoknot partitioning?
So Niels suggesting an experiment. Yes. So in vitro folding, we haven't seen magnesium making a big contribution to this. I showed one little gel where it didn't. That was surprising to me. I mean, we can do this with EDTA and it doesn't seem to make a difference. Inside the cell, I'm not really sure. And, when we add this analog to the cells, we are seeing high, polyamines are high, but I don't know how it changes the distribution. Spermidine in vitro, we can get spermidine at much higher concentrations than spermine to do the folding. So most of our exploration have been, at a few concentrations of these things. But when we have done titrations of the ones that don't look active, we can at higher concentrations get them to do the fold. I don't know if that doesn't answer your question about the in vivo part, but I don't have any information about the in vivo conditions really.
Okay, great. And then maybe we have time for one last. There are more questions, so I think maybe Manny can type answers in response.
I'm happy to answer by email, whatever. Yeah.
Great. So, but just as one final question before we wrap up, slightly different type of question, which is basically, how do you think the pseudoknot directly impacts splicing? Is it occluding a splice site, or like some cis-acting, they have an ESE, an exonic splicing?
Yeah, that's a great question. we haven't really explored that too much. My feeling is that the pseudoknot triggers the formation of kind of a repressive complex. It might include splicing factors or SNRPs, but they're being bound in a way that doesn't promote the assembly of a functional spliceosome. And there's other similar negative splicing regulatory complexes that in other genes that might, be a model for that. The 3' splice site is dangerously close, and we do see changes in accessibility. It could be hiding the 3' splice site in some way. There are proteins known to be important for this splicing event. RBM10, there's a really interesting paper. Oh, geez, now I'm going to forget the author's name. Anyway, dengue virus infection knocks down RBM10, and that leads to activation of a high polyamine state and knocks out SAT's ability to reduce that. And so virus replication requires high polyamines. And so there's some interplay already that we know about between viral infection, a splicing factor RBM10, and the virus's apparent adaptive ability to reconfigure polyamine levels in the cell it's infecting in order to promote its own replication. So there's other stuff that's known about this that I didn't really talk about.
Okay, super interesting. All right, so I'm getting the notification that we should be wrapping it up. I think, thank you so much, Manny, for giving a great talk, and thanks to everyone for, and also thank you to Anca, the first speaker, for giving a really interesting talk, and to everyone for joining us on your late or mid or whatever time of day it might be, wherever you are, to enjoy some really exciting RNA science.